Hello guys, our today's topic is physiology of digestion. So our digestive system is concerned with the digestion of food. So first of all we must know what is food. So food is any substance which provides us materials necessary for growth and development of the body. These materials also sustain our metabolism. So let's define what is food. So food is any substance which provides us materials for growth, development, and metabolism. So any substance which provides us materials for growth, development, and metabolism, that substance is known as food. So mainly consists of our food mainly consists of carbohydrates, proteins, fats, vitamins, minerals, and of course water in it. So the foods that we consume, they mostly contain carbohydrates, proteins, fats, vitamins, minerals, and water in it. Although all the foods may not contain all these items together, but mostly our foods, either they are rich in carbohydrates, they are rich in proteins, or they may be rich in fats. Some foods are rich in vitamins and minerals. And almost all the foods contain water in it, right? So out of these five nutrients, carbohydrates, proteins, fats, vitamins, and minerals, out of these five nutrients, only three nutrients, they require digestion. They require digestion. While these materials, water, minerals, and vitamins, they do not require digestion. Don't require. They do not require digestion. They are absorbed as such from the alimentary canal. So the components of our food, carbohydrates, proteins, fats, they require digestion. Why they require digestion? Because they are cellular macromolecules. They are large molecules. While as vitamins, minerals, and water, they do not require digestion. Why? Because they are small molecules, right? So we will see how these carbohydrates, proteins, and fats are digested in our alimentary canal. So let us begin with the digestion of carbohydrates first, right? So let us see the digestion of carbohydrates. So first we will discuss digestion of carbohydrates. We take carbohydrates mostly in the form of starch. We take carbohydrates mostly in the form of starch. You know what is starch? So starch is the stored carbohydrate of plants. So when we take carbohydrates in the form of starch, their digestion starts right from the mouth. So digestion of carbohydrates, it starts from the mouth. Those enzymes which are involved in digestion of carbohydrates or starches, those enzymes are generally known as amylases. Enzymes which are involved in digestion of carbohydrates or starches, they are generally known as amylases or sometimes we also call them as carbohydrolysis. Carbohydrolysis. So those enzymes which are generally involved in digestion of carbohydrates and starches, they are known as amylases or carbohydrolysis. So digestion of carbohydrates, it starts from mouth. So when we ingest the food into our mouth, this food, it contains some starch. Starch may be in the form of plant starch or it can be in the form of animal starch that is known as glycogen, right? So when we ingest carbohydrates in the form of plant starch or animal starch glycogen, in our mouth it is mixed with the saliva. In saliva we have got salivary amylase. We have got ptyalin. Ptyalin. It is having salivary amylase. Salivary amylase. So this salivary amylase, it acts on the starch and glycogen in our mouth and it breaks the starch and glycogen into oligosaccharides like maltose plus isomaltose, and small sugars known as limit dextrin. Limit dextrin. So as we ingest the carbohydrate rich food into our mouth, we start masticating this food. In the process of mastication, we mix saliva with our food. This saliva, it contains an enzyme, salivary amylase in it. And this salivary amylase, it breaks this starch or glycogen into disaccharides like maltose, isomaltose and limidextrins. We generally call them as oligosaccharides, right? So it breaks them into oligosaccharides like maltose, isomaltose and limidextrins. This salivary amylase, it works in slightly acidic medium, 6.8 pH. In our mouth, we have got slightly acidic pH, right? So at a pH of 6.8, this salivary amylase of saliva, it converts starches and glycogens into maltose, isomaltose, and limit dextrins, right? If we masticate the food properly in our mouth, if we properly masticate food in our mouth, 30% of the starches, 30% of starch, they may be converted into maltose, isomaltose, and limit dextrins only in our mouth, right? So as we are taking food, we should properly masticate the food because proper mastication of food helps us in digestion of starch and glycogen. It helps us in digestion of carbohydrates. So if we properly masticate the food, approximately 30% starch may be converted into maltose, isomaltose, and limit dextrins, right? So this is the action of salivary amylase on starches and glycogen in our mouth. Now, after proper mastication, we swallow the food and this food goes into stomach. In stomach, gastric juice is produced. In stomach, gastric juice is secreted. In this gastric juice, there is no there is no amylase present. Gastric juice contains no amylase. In gastric juice, there is no amylase present, right? So gastric juice doesn't help us in the digestion of starch. Gastric juice doesn't act on starch. However, the HCl present in gastric juice that helps us in softening up the plant food material which we ingest. So stomach secretes gastric juice. In that gastric juice, there is no amylase present. So gastric juice doesn't help us directly in the digestion of starches or carbohydrates, right? But still gastric juice helps us in a different way. How? Gastric juice contains HCl, and this HCl, it softens the plant materials. So when we are taking starch, when we are taking starch in the form of plant foods, right? So that starch contains cellulose fibers in it that contains cell walls in it. So it is the HCl of gastric juice which breaches those cell walls, which breaks those cell walls, releasing the cytoplasmic content for further digestion, right? So in stomach, there is no direct digestion of amylases, the starches or glycogen. However, the HCl of gastric juice help us in softening the plant materials which we consume. It causes breaches in the cell walls of the plant materials, so that help us in the digestion. But it doesn't have any amylase in it. Now, after stomach, the food goes into small intestine. As the food mixes with the gastric juice, gastric juice, this food becomes now acidic, and this acidic food, which leaves the stomach and enters into small intestine, that acidic food is known as chyme. That acidic food is now known as chyme. So this chyme will now enter into small intestine. So chyme is the acidic food which leaves the stomach. Acidic, partially digested food which leaves stomach to enter into intestine. So chyme is the acidic partially digested food which leaves the stomach to enter into intestine for further digestion. So as the chyme enters into small intestine, in this small intestine, it comes in contact with, first it comes in contact with the pancreatic amylase. So after proper mastication of food, 30% of the starches are digested in the mouth. But as the food enters into the small intestine, the first part of small intestine is duodenum. You know, in duodenum, hepatopancreatic duct opens, and that contains pancreatic juice. So pancreatic juice of pancreas, it acts on starch plus glycogen. It acts on starch and glycogen, and this pancreatic amylase, it breaks the starch and glycogen further into maltose, isomaltose plus limit dextrin. 30% of this starch and glycogen was digested in mouth. Now the remaining 70% of the starch and glycogen, it is acted upon by pancreatic amylase. And pancreatic amylase also converts the starch plus glycogen into maltose, isomaltose, and limit dextrins. Now the food comes in contact with the intestinal juice. Now food comes in contact with intestinal juice. In intestinal juice, we have starch digesting carbohydrate digesting enzymes known as oligosaccharides. Oligosaccharides. In the intestinal juice, we have got starch digesting enzymes known as oligosaccharides. So they will digest these oligosaccharides, which were formed by the action of salivary amylase and pancreatic amylase. These are all known as oligosaccharides. They are all known as oligosaccharides, right? So in intestinal juice, first of all, maltose is acted upon by. Maltose is acted upon by an oligosaccharide known as maltase. So maltase of intestinal juice, it converts maltose into 2-glucose. 2-glucose. You can see here Maltose is a disaccharide, right? Similarly, isomaltose. Isomaltose. It is acted upon by another oligosaccharide known as isomaltase. Isomaltase. And it breaks it into two glucose units. Then limit dextrins. Limit dextrins. Limit dextrins means small sugars. So these limit dextrins, they are acted upon by enzyme limit dextrinase, and it converts it into glucose. The number of glucose units may vary. Sometimes there may be two, sometimes there may be three, sometimes there may be four. So these small sugars, they are known as limit dextrins, and they are acted upon by another enzyme of intestinal juice, limit dextrinase, and it converts these limit dextrins into glucose units. Similarly, we take sugar along with tea, coffee, sweets, etc. So that sugar which we consume along with tea, coffee, sweets, etc. That is sucrose. Sucrose, it is also known as cane sugar, right? So this sucrose, it is acted upon by an intestinal oligosaccharide known as sucrase. So this sucrase, it breaks sucrose into glucose plus fructose. It breaks sucrose into glucose plus fructose. Similarly, we consume milk. Milk contains milk sugar, lactose. So this lactose of milk, it is acted upon by intestinal oligosaccharides known as lactase. And this lactase of intestinal juice, it breaks lactose into glucose plus galactose. So in this way, all the carbohydrates, all the starches which we had consumed are finally converted into monosaccharides. The most abundant monosaccharide being glucose. In addition to glucose, other monosaccharides may be released, like fructose, galactose, right? So at the end, monosaccharides are produced. So what is the end product of starch digestion in our body? So the end product of starch digestion in our body is monosaccharides. End product of starch digestion in our body are monosaccharides. The most abundant monosaccharide is glucose, right? So in this way, all the dietary carbohydrates which we had consumed are finally converted into monosaccharides, right? So that is all about the digestion of carbohydrates in our body. Now, after this digestion of carbohydrates, we will now discuss digestion of proteins. Digestion of proteins. So enzymes which are responsible for protein digestion in our alimentary canal are generally known as proteases. They are generally known as proteases. So these proteases, they are of two types. One class of proteases is known as endopeptides. And another class of proteases is known as exopeptides. So we know proteins, they are formed of long polypeptide chains. They are formed of polypeptide chains. So we need to break these polypeptide chains. For breaking these polypeptide chains, we have got proteases, which are of two types: endopeptidases and exopeptidases. So what are endopeptidases? These endopeptidases are the enzymes which break internal peptide bond of a polypeptide chain. Suppose this is a polypeptide chain. amino acid, amino acid. So this is the polypeptide chain. This is the long polypeptide chain. This is the long polypeptide chain, right? So endopeptidases, they cut this long polypeptide chain by breaking an internal peptide bond. Suppose it breaks the peptide bond here. It breaks the peptide bond here. And it will divide this long polypeptide chain into two short fragments. So endopeptidases are those peptidases or those protein digesting enzymes which break an internal peptide bond of a long polypeptide chain. So it breaks the internal peptide bond of a polypeptide chain, and this polypeptide chain is broken into two small fragments. These fragments of polypeptide chain, they are known as peptones and proteoses. They are known as peptones or proteoses, or we simply call them as peptides. So endopeptidases are those peptides, proteases. Endopeptidases are those proteases, protein digesting enzymes, which break which break internal peptide bond of a long polypeptide chain, dividing the polypeptide chain into two short fragments known as peptones and proteoses, which are together known as peptides. So they break polypeptides into small peptides. This is endopeptidases. Then we have got exopeptidases. Then we have got exopeptidases. Now these exopeptidases, they break the terminal peptide bonds of a polypeptide chain. They may break the terminal peptide bond of a polypeptide chain. They break terminal peptide bonds of a polypeptide chain. So this is one terminal peptide here. This is another terminal peptide here. This is another terminal peptide bond here. This is terminal peptide bond here, right? So they cut the peptide bonds from the terminal side of a polypeptide chain or a peptide chain, right? That is why we call them as exopeptides, peptidases. Exo means external, endo means internal. Endo means internal, exo means external. So they attack internal peptide bonds. They attack external peptide bonds. So this is the external most peptide bond. It is attacked. This is the external most peptide bond. This is attacked. This is the external most peptide bond. It is attacked. This is the external most peptide bond. It is attacked. Right? So those proteases which break terminal peptide bond of a polypeptide chain, they are known as exopeptidases. The examples of endopeptidases are pepsin, pepsin, trypsin. These are examples of endopeptidases. Pepsin and trypsin, they are endopeptidases. The examples of exopeptidases, they include exopeptidases, they include enzymes like carboxypeptidase, aminopeptidase, aminopeptidases, right? Or these are the examples of exopeptidases. Carboxypeptidase and aminopeptidase. They are examples of exopeptidases. So before we start the digestion of proteins, we have to keep it in mind that protein digesting enzymes, they are known as proteases. And we generally classify proteases into two types, endopeptidases and exopeptidases. Endopeptidases are those proteases which break the internal peptide bond of a polypeptide chain, dividing it into two short fragments known as peptones and proteoses, or we generally call these two short fragments of polypeptides. We call them as peptides. Exopeptidases are those proteases which break the terminal peptide bonds of these peptides, which were formed by endopeptidases. So first endopeptidases will cut the protein into smaller fragments known as peptides, right? Then exopeptidases, they will act on these peptides, proteoses and peptones. They will break their terminal peptide bonds to release individual amino acids, one by one. So first this bond will be broken. Then second, this bond will be broken. Similarly, from this end, first this bond will be broken. Then second bond will be broken. So they act stepwise, one by one. They break first peptide bond here. They break peptide bond here. They break next peptide bond here. They break next peptide bond here. Then another peptide bond is broken. Another peptide bond is broken. Right. So they stepwise release amino acids. They release amino acids and dipeptides at the end. And dipeptides at the end. So the endopeptidases, they break proteins into short protein, short peptide chains known as peptides, proteases and peptones. Exopeptidases, they break these proteases and peptones. They break peptides into amino acids and release at the end dipeptides. So this is the two types of proteases, endopeptidases and exopeptidases. Now this protein digestion, it starts from stomach. In mouth, saliva is produced. In that saliva, there is no protein digesting. So saliva doesn't contain any protein digesting enzyme. So digestion of proteins, it starts in the stomach. You know, in stomach, gastric glands, they release gastric juice. How much gastric juice is released per day? 2 to 3 liters of gastric juice is released per day into the lumen of stomach. Approximately 2 to 3 liters of gastric juice is released per day into the lumen of stomach. So we will see how these proteins are digested in stomach. So in stomach, gastric juice is released. In this gastric juice, we have got HCl. In this gastric juice, there is HCl. There are proenzymes. In gastric juice, we have got proenzymes. There are two proenzymes, pepsinogen and prorin. Right? And this gastric juice also contains a gastric lipase. Gastric lipase as well. So protein digesting enzymes, pepsinogen and prorin, they will only act on the proteins by the help of this HCl. So in stomach, first these proenzymes, these two proenzymes, they need to be activated. When the food, protein containing food enters into stomach, these two proenzymes, pepsinogen and prorin, they need to be activated. So this pepsinogen, it is activated by HCl, at a pH of 1.2 to 1.8. And this pepsinogen is converted into pepsin. It is converted into pepsin. Similarly, in children, prorenin, it is also activated by HCl at a pH of 1.2 to 1.8. And it is activated to renin. So when we consume proteins, gastric juice is released. Gastric juice contains some enzymes in it, but those enzymes, they are in inactive form. Those inactive enzymes, they are known as proenzymes. So first of all, we need to activate these proenzymes. So how these proenzymes are activated in our stomach? These proenzymes are activated in our stomach by the help of HCl. So due to a presence of HCl, strong acidic conditions are created there. And at a pH of 1.2 to 1.8, this HCl converts pepsinogen into pepsin, the active enzyme, and prorenin into renin, an active enzyme. Now this pepsin, it acts on the proteins. It acts on the proteins which we have consumed. This pepsin breaks these proteins. Pepsin is an endopeptidase. So it breaks them into peptones and proteoses, which we collectively call as peptides. So in stomach, proteins are attacked by pepsin, and pepsin converts these proteins into peptones and proteoses, collectively known as peptides. Right? Similarly, when an infant takes the milk, renin is only secreted in infants. As the infants grow up, then renin production stops. In adults, there is no renin secreted in the stomach. So renin is only secreted in the infants. As adults, we don't have any renin in our stomach. So this renin, it acts on the milk protein. When we consume milk, in that milk there is a protein known as casein. Casein protein. This casein of milk, it is acted upon by renin. It is acted upon by renin. And renin converts this casein into paracasein. Then this paracasein, it gets precipitated in the presence of calcium salts. In the presence of calcium salts, it gets precipitated and it changes into calcium paracasein. It changes into calcium paracasein. That calcium paracasein is also known as curd. So it changes into curd, right? Calcium paracasein. Then this calcium paracasein, it is acted upon by pepsin. And pepsin breaks it into peptones and proteoses. So we call they are changed into peptides. So this is how proteins are digested in our stomach. So when proteins reach to the stomach, in the stomach these proteins are first acted upon by pepsin. Pepsin converts them into peptones and proteoses, collectively known as peptides. Similarly, when infants take milk, that milk contains the milk protein casein. That casein is acted upon by renin, and renin converts it into first paracasein, then paracasein in presence of calcium salts, which are present there in the milk. It gets precipitated as calcium paracaseinate. So this calcium paracaseinate is known as curd, right? That is why you might have seen in most of the infants, this milk is digested out, it is vomited out. And this milk is mostly in the form of curd. That milk which infants vomit out is mostly in the form of curd. Why do infants vomit out that milk? Because their cardiac sphincter is not fully functional yet. Their cardiac sphincter is not fully working. So for that reason, some of the gastric content, some of the curd that is vomited out, right? If any infant vomits out uncurdled milk, so that indicates a gastric problem. If an infant vomits out uncurdled milk, that indicates the gastric problem. If curdled milk is vomited out, that is normal, right? So milk protein casein, it is converted into paracasein. Then paracasein precipitates as calcium paracasinate. Then this calcium paracasinate is acted upon by pepsin, and it is converted into peptones and proteoses that we call as peptides, right? So this is digestion of proteins in stomach. Now from the stomach, these proteins, they now go into the small intestine. Now from stomach, these proteins they enter into intestine. In intestines, different juices are secreted. Intestines they receive first bile from liver. Intestines receive pancreatic juice from pancreas, and intestines also receive intestinal juice from the wall of small intestine. Bile does not contain any digestive enzyme. No enzyme is present in bile. Bile doesn't have any digestive enzyme in it. Then what is the function of bile? As I told you just now, the acidic food which leaves the stomach, that is known as chyme. So when this chyme enters into small intestine, in small intestine the enzymes work in alkaline medium. So this acidity of the chyme, it must be neutralized first. So bile helps us in the neutralization of acidity of the food. Bile helps us in neutralization of acidity of food. It helps us in neutralization of acidity of food. Acidity of food. And this bile, it also creates alkaline medium. Alkaline medium in small intestine. In small intestine for intestinal enzymes. For intestinal enzymes to work properly. For intestinal enzymes to work properly, right? So the function of bile is first to neutralize the acidity of food. It creates alkaline medium for in small intestine for intestinal enzymes to work properly. And you also know this bile is involved in emulsification of food. It is essential for the digestion of fats. It first emulsifies the fats. It is involved in the emulsification of fats. Without emulsification of fats cannot be digested properly, right? So it has got these roles, right? So leaving the bile aside now, in intestine, we have got pancreatic juice and we have got intestinal. Now we will see first which enzymes are present in pancreatic juice, which enzymes are present, which protein digesting enzymes are present in intestinal juice, right? In pancreatic juice, there are some protein digesting enzymes present. For example, in pancreatic juice, we have got trypsinogen. Trypsinogen. It is a protein digesting enzyme. In pancreatic juice, we have got chymotrypsinogen. Chymotrypsinogen. It is a protein digesting enzyme. And in pancreatic juice, we have got procarboxypeptidase. Procarboxypeptidase. This is also a protein digesting enzyme, right? So these are the enzymes which are present in the pancreatic, which are secreted by pancreas. These are all protein digesting enzymes. Trypsinogen, chymotrypsinogen, and procarboxypeptidase. Similarly, in intestinal juice, there are some enzymes present. In intestinal juice, there is an enzyme that is known as enterokinase. Enterokinase. Then there is another enzyme that is known as aminopeptidase. Aminopeptidase. And then there is another enzyme present in intestinal juice, which is known as dipeptides. Dipeptides. So these are the enzymes, protein digesting enzymes, which are present in the pancreatic juice and intestinal. So as the protein is reached into small intestine, it mixes with the pancreatic juice and intestinal juice. In pancreatic juice, there are some protein digesting enzymes, trypsinogen, chymotrypsinogen, and procarboxypeptidase. In intestinal juice, there are some protein digesting enzymes, enterokinase, aminopeptidase, and dipeptidase, right? So we will see how they will work. All these enzymes, which are present in the pancreatic juice, they are all proenzymes. They are all proenzymes. Proenzymes means they are all inactive enzymes. So for protein digestion, they must be first activated. They must be first activated. So first of all, trypsinogen is activated. Trypsinogen, it is activated. What activates this trypsinogen? This enterokinase of intestinal juice. Enterokinase of intestinal juice. It activates trypsinogen into trypsin. So it carries out the catalysis of autocatalysis of trypsinogen into trypsin. This enterokinase is the only protein digesting enzyme which does not directly digest the proteins. It helps us in the digestion of proteins indirectly. So how it indirectly helps us in the digestion of proteins? It is not directly involved in digestion of protein. This trypsinogen, which will be converted to trypsin, it will directly involved in protein digestion. Chymotrypsin, procarboxypeptidase, aminopeptidase, dipeptidase, they are directly involved in protein digestion. But enterokinase is not directly involved in protein digestion. Then what is its role? It is only involved in conversion of trypsinogen into trypsin. So it carries out the autocatalysis of trypsinogen into trypsin in an alkaline medium, 8.0 to 8.8 pH. Right? So when trypsin is formed, when trypsin is when trypsinogen is converted into trypsin, then this trypsin it stimulates, it activates other enzymes. Now this trypsin, it will change chymotrypsinogen into chymotrypsin, into chymotrypsin. Similarly, it will change procarboxypeptidase, procarboxypeptidase into carboxypeptidase, into carboxypeptidase, right? So, first of all, trypsinogen is converted into trypsin by this enterokinase. Once the trypsin is formed, then this trypsin stimulates, it activates other enzymes. It activates chymotrypsinogen into chymotrypsin. It activates procarboxypeptidase into carboxypeptidase. So in this way, the enzymes are first activated for carrying out protein digestion. Now we will see how these enzymes will work, how they will digest the protein. For that, I have to rub this board first. Now those proteins which were not digested in stomach, those proteins are acted upon by. Those proteins which were not digested by this pepsin in stomach, as they reach into small intestine, those proteins are here acted upon by trypsin. Trypsin is an endopeptidase, and it converts them into peptones and proteoses, collectively known as peptides. So protein digested started in stomach. So some of the proteins they were digested in stomach by the action of pepsin. But pepsin could not digest all the proteins. So some of the proteins they remained undigested in the stomach. Now as those proteins reach into small intestine, they are acted upon by trypsin. And trypsin being an endopeptidase, it converts these proteins into peptones and proteoses, known as peptides. Similarly, as I told you, in adults, renin is not secreted in gastrin. Then how do adults digest their milk? Adults also consume milk, right? Then how we digest our milk? So in adults, this milk protein, casein, it is acted upon by it is acted upon by chymotrypsin. It is acted upon by chymotrypsin in small intestine. And this chymotrypsin converts it into paracasein. Then this paracasein in presence of calcium salts. Then this paracasein in presence of calcium salts. It changes into calcium paracasein. Calcium paracasein. Paracasein. Or we call it as curd. Now this curd, which is formed in our small intestine, it is acted upon by trypsin. And trypsin breaks it into peptones and proteoses. Proteoses, known as peptides. Now trypsin breaks it into peptones and proteoses known as peptides. So in adult animals, milk is not digested in stomach. So milk is digested in their small intestine. So milk protein casein in small intestine, it is acted upon by chymotrypsin. And chymotrypsin converts it into paracasein. Then this paracasein gets precipitated as calcium paracasein. Then this calcium paracasein it is acted upon by trypsin. And trypsin converts it into peptones and proteoses. Right? Now all the dietary proteins, they have been converted into peptones and proteoses. So all the proteins which we had consumed are now converted into peptones and proteoses or peptides. Now these peptides they are acted upon by the aminopeptidase and carboxypeptidase. Now these peptides they are acted upon by aminopeptidase, aminopeptidase and carboxypeptidase simultaneously, and they release amino acids plus dipeptides at the end. So peptides they are acted upon by peptidases, aminopeptidases and carboxypeptidases, and they release amino acids, and at the end a dipeptide is released. Right. So how do these aminopeptidases and carboxypeptidases work? As I told you, an a polypeptide chain has got two ends. Suppose this is a polypeptide chain. This is a polypeptide chain. Suppose this is a polypeptide chain. This is a polypeptide chain. So this polypeptide chain, it has got, or this peptide chain, it has got two ends. On the one side, it has got a free amino group. At the another terminus, it has got a free carboxyl group. So this peptide chain, it has got two ends. On one end, it has got a free amino group. At the other end, it has got a carboxyl group. So this end of this peptide chain, which is having free amino group, it is known as N terminus. It is known as N terminus. And this side of the peptide chain, which is having carboxyl group, it is known as C terminus. It is known as C terminus. So aminopeptidases, they act on the side of N terminus. They act on N terminus. And carboxypeptidases, they act on C terminus. So aminopeptidases, they will break the first peptide bond from the N terminus. That means they will break this peptide bond, releasing this amino acid. Carboxypeptidase, they will break this peptide bond, releasing the amino acid. Right? So in this way, one by one, aminopeptidases will act from N terminus. Carboxypeptidases will act on C terminus, breaking the peptide bonds one by one, releasing one amino acid, another amino acid, another amino acid, another amino acid, and finally releasing a dipeptide. These are two amino acids linked together, and they are known as a dipeptide. This dipeptide, it will be then digested by dipeptidase enzyme. So all those peptides which were formed by the action of endopeptidases are now digested by exopeptidases. Aminopeptidase, carboxypeptidase, they are exopeptidases. Aminopeptidase acts on N terminus. Carboxypeptidase acts on C terminus of a peptide chain, releasing one amino acid, another amino acid, another amino acid, another amino acid, and at the end, giving rise to a dipeptide. So this is the action of aminopeptidases and carboxypeptidases. Now what remains there? Now only dipeptides are there. These dipeptidases, they are acted upon by the dipeptidase enzyme. Dipeptides, they are acted upon by dipeptidase. Dipeptidase enzyme is present in intestinal juice. It breaks this dipeptide into two amino acids. Right? So this dipeptide, when the peptide bond will be broken, one amino acid will be released, another amino acid will be released, right? So dipeptidases, they break this peptide bond between two amino acids. Dipeptides, they break this peptide bond between two amino acids, releasing then two amino acids. So when this peptide bond is broken, this amino acid is released and this amino acid is released. So two amino acids are released. So in this way, end product of protein digestion is amino acids. Amino acids. So all the proteins which we consume in our food, those proteins will be finally converted into amino acids in a stepwise manner, first in stomach, then in intestine, by the help of different enzymes, and at the end, amino acids will be produced. So that is all about the digestion of proteins. Thank you.